Welcome to the Neuroendocrine Cancer UK Q&A session on the subject of genomics. This series of Q&A sessions has been created as part of the Neuroendocrine Cancer UK Virtual Patient Handbook, which is due to be released in 2023. My name is Beth, and I'll be hosting these sessions with various medical experts, covering a range of questions within neuroendocrine cancer care. Although I have worked with Neuroendocrine Cancer UK for a little while now, I'm not a medical professional myself, and so in many senses, I'm learning alongside you, the audience. Some of the questions I'll be asking have been taken directly from the neuroendocrine cancer community, and the focus of these sessions will be on providing useful information and advice in a concise audiovisual format. In this session, Professor Chrissie Thirlwell will be answering a range of questions on genomics. But firstly, let's get to know her a little better.
Hello, hi, I'm Chrissie Thirlwell. I'm a medical oncologist. I've been working in the field of neuroendocrine tumours for 15 years or so now. I also spend quite a lot of time doing research, and I look at DNA sequencing in cancer, but particularly in neuroendocrine tumours. And I've been trying to learn as much as possible to help us identify new treatments and identify how and where neuroendocrine tumours come from over the last 10 or 15 years or so. Alongside that, I'm also the secretary of the UK NET Society, and I'm co-chair of the Board of Scientific Advisors for the NET Research Foundation in the USA.
Great, thank you. Thanks, Chrissie. So firstly, just to say, I was watching your 2020 summit video for Neuroendocrine Cancer UK recently, and in that you gave a really good Genomics 101 at the start. So I'll include a link to that video in the upcoming virtual patient handbook, which this Q&A session is being made for. So if people are interested, they can kind of learn more from that video. And actually in this Q&A session, I'm going to be asking you a few kind of follow-on questions from that summit video. But just to introduce my first question, I was interested in the explanation you gave around germline mutations, and you mentioned some inherited syndromes such as multiple endocrine neoplasia, among others. So to begin, can you explain a bit about whether neuroendocrine cancer is hereditary?
Thank you. So we know that around 10 to 15% of neuroendocrine cancer is hereditary, and there are these syndromes that you've mentioned which are aligned with certain types of neuroendocrine cancer development. So there's the multiple endocrine neoplasia 1 and MEN2, so shortened to MEN1 and 2. When we look at the different types of neuroendocrine cancer, we know that MEN1, for example, can lead to some tumours in the lung, so bronchial neuroendocrine tumours, and also the pancreas. But of the most common types of neuroendocrine cancer, so we think about the lung, the pancreas, and the small intestine, it's mainly the pancreas where there seem to be a higher incidence of these germline inherited mutations, which might lead to that cancer development. So again, pancreatic neuroendocrine tumours are associated with MEN1, but you can also have von Hippel-Lindau VHL or NF neurofibromatosis NF1 mutations as well. And some really interesting work about 5 years ago now, looking at the whole genome sequencing in the germline, identified around 17% of pancreatic neuroendocrine tumour patients might have a mutation, a germline mutation. So some new genes were identified there as well. When we think about the small intestinal nets, one of the most common types of nets, there really aren't any inherited syndromes that we know of. And I've been focusing on working in that area for quite some time. So there aren't syndromes or genes that we would automatically look for, but there still is a concerted effort to really understand how these tumours develop. And there's some families where we know that more than one person might have a small intestinal net. So that gives us a hint there might be something going on, but we haven't actually identified the genes for small intestinal nets yet.
Okay, thank you. So on to the next question. As the lead for the Neuroendocrine Tumour 100,000 Genomes, the GSIP, so Genomics England Clinical Interpretation Partnership, I was wondering if you could provide any updates on our understanding of the impact of the 100,000 Genomes project now that we're around 5 years from its completion.
Thank you. So alongside leading the neuroendocrine tumour GSIP, I've also been looking and leading on the cancer arm level, so across all of the cancers that were sequenced. And this project, and if you just Google 100,000 Genomes Project, you can get more information easily through Genomics England. 17,000 cancers underwent whole genome sequencing. It's the largest cohort globally, the most phenomenal piece of work that was only made possible by the infrastructure of the NHS in the UK. So that was absolutely phenomenal. Within that, and representing perhaps the rarity of neuroendocrine tumours, only around 130 net cases were in that 17,000 cancers. With the more common cancers such as bowel cancer and breast cancer, there are a few thousand in there. But that doesn't mean, from looking at the wider picture across cancer, it's not that we can't then learn things that are relevant to neuroendocrine tumours. And the biggest finding across all of those 17,000 cancers is around 50% of them identified some kind of change in the DNA in the tumour, which might either identify a target, and I know we're coming on to personalised medicine, so a target for a personalised treatment, or maybe a target that would give someone access to a clinical trial. And that data was really, really powerful, which then led to cancer being a big part of the genomic medicine service, which I'm now part of, and I can talk about a bit later. When we think about the neuroendocrine tumour GSIP with these 130 cases, my work with my group, but also others internationally, we know already before the 100,000 Genomes Project, we don't have these actionable mutations. We don't have common mutations in neuroendocrine cancers, which we can give treatments to. And the background mutation rate in these tumours is very low, so they don't behave like other cancers. They're really unique and they're different. They're intriguing, but it's a real challenge when we're trying to work out what makes them tick and what makes them develop. We do see mutations in the pancreatic and the bronchial neuroendocrine tumours, which actually are related to how our DNA gets wound up and packaged when the cells divide and unpack. That's really interesting, but none of those genes are actually what we call targetable. So we don't give a medicine to target those. So what we found so far in our analysis of the neuroendocrine tumour GSIP is we found some similar findings to what my group has found previously, or other groups internationally have found. And because of that, we're focusing on epigenetic changes now. We've got a piece of ongoing work looking at the DNA methylation and using some novel sequencing approaches for that across the whole of the genome. So in the last couple of years, there's really, really new and exciting type of sequencing of the whole genome called long read sequencing. So we're planning to apply that to our neuroendocrine tumour GSIP. So that's coming now. So we've looked at all of the regular sequencing. We've identified some of the mutations that we've known about before, but unfortunately we haven't found here are 3 or 4 brand new things. But we're now focusing our efforts on looking at novel sequencing approaches alongside that, and also the DNA methylation, which I think I mentioned quite a lot in the summit talk. If you wanted to look at that again.
Great. And actually, when I was researching the project, I saw that the government announced in 2018 that over the following 5 years that they would hope to sequence 5 million genomes. So was this target achieved, or is it still an ongoing goal?
I think the most impactful thing that has come from that piece of work is the NHS Genomic Medicine Service, which in the Southwest, where I'm based and work, I'm the clinical director for the Southwest Genomic Medicine Service Alliance. And we're the first country globally to roll out the application of genomics in medicine across the board. So this encompasses not just cancer, but it encompasses all cancer and lots of rare disease, lots of looking at how your DNA affects how you handle drugs. And there's also a really novel piece of work around sequencing babies and then using that information through life. So that's something that's coming on in the next year or so. So I think, it's one of the most forward-thinking, transformative services that I've seen in the NHS in my in my career to date, which is why I got involved. And in terms of the cancer, what these 5 million genomes look like, and that's across everything. So it's not 5 million cancers. So cancer's part of that, a big part of that. And the way that most common cancers, so breast, lung, bowel, and other cancers are approached is we have a 500 gene panel. So instead of doing the whole genome sequencing as in the 100,000 genome project, you do a panel when that has all the usual suspects and you can get information really quite quickly. And that's available for pretty much all what we call solid cancers already through the Genomic Medicine Service. With respect to neuroendocrine tumours, as I've mentioned, they don't have these usual suspects in terms of the genes that might be on the panel that we can give medicines to, but there's still opportunities in the Genomic Medicine Service, both for what we call the tumour agnostic therapies. So I think your interviewer Deb Sarkar highlighted that. So for example, in NETs, there's a NTRK, which is something which is what we call tumour agnostic. So there's access to sequencing for that, and something for all rarer cancers, and neuroendocrine tumours are included in that. Once an individual has reached the end of the standard lines of therapy that we might give in clinic, their tumour can undergo sequencing to see if there is something identified there which could then be targeted. The only caveat I would say around that approach is that, if it identifies a gene that's perhaps targeted, so EGFR, it's just a random gene that we can target in lung cancer, for example. There are drugs which target that. That doesn't necessarily mean that we can just grab that medicine off the shelf and give it to a patient. Obviously, we have to be mindful and safety. There's licensing and there's laws and legality around how we use medicines. So the sequencing can be very helpful, but sometimes we might then, as clinicians, have conversations with the companies that provide that drug to see if they can provide that, to see if it can help that individual. So that's the sort of the kind of steps that we're getting to.
Leading on to the third question about personalised medicine, so you touched a little bit there about one of our other Q&A sessions with Dr. Deb Sarkar. He mentioned a bit about the work being done in gene sequencing and, like you say, tumour agnostic treatments. This seems to be a really exciting field of study, and I wondered if you could talk a bit about that in the context of personalised medicine more generally. We hear personalised medicine talked about, I think, quite a bit. And, are people experiencing it already in the NHS, and how does that look like and for them?
Yeah, the short answer is yes, definitely. And the treatment of cancer has been at the forefront of using personalised treatments to target molecular pathways or drivers in tumours, which are causing them to grow and spread. So for some years now, initially through clinical trials, and we'll come on to that, we've identified there might be a particular change in a protein or a target in a tumour, which we have a drug which is specifically aligned to, which would treat that we know there's that change in that specific tumour. That was over the last 10 or 15 years or so, there's been thousands and thousands of trials and studies looking at different targets and personalised approaches to treating different cancers. And we now use this routinely in standard of care in oncology in the NHS. Where some of the challenges with personalised medicine in cancer is, again, through DNA sequencing, we've learned that even within one tumour, so this is one tumour, different areas of that tumour might harbour one of these molecular changes and other areas don't. So if that's called heterogeneity, so we call it tumour heterogeneity, and also we have these clones, and that's what clonal evolution. So if you've been reading about that, heterogeneity and clonal evolution is a really big challenge to us with molecular target therapy. So if I've got my tumour here, and I've got something that targets that bit, if the rest of the tumour doesn't contain that target, then that will continue to grow. So you can sometimes see some response, but then unfortunately other areas of the cancer continue to grow, or you can actually treat the whole cancer, but then it evolves. they're really wily cancers. They evolve and they adapt and they change. And you may have seen there's recently press coverage last week around a massive study in lung cancer called TracerX. That's Professor Charlie Swanton, the CRUK. Lots of news coverage because it's been really intricate, detailed sequencing around different lung cancer all over the body, and it's mapped out how the tumors can adapt to change, evolve. And so the biggest challenge for us as cancer doctors is we're chasing a moving target, and that applies to all cancers. So for some cancers already there are molecular targets, very clear personalized medicine, all as standard lines of therapy. But for other cancers, this is still part of clinical trials and we're still investigating new targets, or we might be looking at using two therapies together, or a molecularly targeted therapy with immunotherapy. So there's different combinations that we look at. And where we are with neuroendocrine tumors, again, it is really frustrating and it is really challenging because we don't have any of those obvious usual suspects that we can target with personalized medicine. There are some of the tumor agnostic, as we mentioned, the NTRK. But there aren't sort of the standard off-the-shelf molecularly targeted therapies that we use based on sequencing. But we know, for example, through clinical trials that medicines such as sunitinib and everolimus can be used in pancreatic neuroendocrine tumors, and everolimus can be used in pancreatic and bronchial and intestinal. So we know they are targeted therapies, but we don't make those clinical decisions based on sequencing. That was from the output of clinical trials. So there are subgroups of neuroendocrine tumors that we use different treatments for, but that's mainly historically from really large scale international clinical trial work rather than the DNA sequencing that I talked about earlier.
Okay, thank you. And then as a follow on to that, back to the 2020 summit again, the video that you did for us. In that you talk a bit about the NEN genome sequencing and how it's interesting that the background mutation rate for NETs is very low. You say you're still researching it as you don't understand why it's so low. I just wondered if there was any more insights into that since then.
No, so some of the work that I've been doing with international collaborators in the States and Switzerland and Spain and other countries, because when we're researching rarer diseases and rare cancer, you have to have big international efforts so you can get the numbers and you can get the momentum to do the work. So since 2020, some of the areas where we've been progressing is with whole genome sequencing in mainly in small intestinal nets, and also looking at sequencing for looking at DNA methylation and seeing what we can find from that. So some of the work we've been doing in small intestinal nets, there's more than one primary called multifocal primaries. You can have more than one original tumor dotted along the small bowel. And we can see that in up to 50% of cases. So we've been studying those individual primary tumors to see if they're different, see if they come from the same clone background or different clones. And we've proven definitively they're from different clones and backgrounds. And looking at the methylation of that, the DNA methylation, we can use that to apply clocks to see how fast they might be turning over, or to see how long they may have been around. So we've been applying these epigenetic clocks to those multifocal tumors, which again shows that they've popped up at different times, whereas historically it was thought they'd all come from the same origin. So we've thrown over that theory that they're not clonal in origin, which again tells us a bit more about where they might come from, in terms of there might be something affecting an area of the bowel which then predisposes these tumors to arising. And we've looked very, very closely to see if there are any germline mutations in these individuals, and there aren't. Because if you think of other cancer types, we often look at other cancers and see what lessons we can learn from that. So sometimes with germline inherited cancer predisposition, you might get lots and lots of different primaries because you've got this background sort of change or error going on. But again, we don't see that in the germline. So we're learning more about the biology, particularly where these multifocal primaries arise from. And then most recently we've been looking at applying novel sequencing approaches. So there's something called Oxford Nanopore long read sequencing, where you just have I mentioned we have your 3 billion bases of DNA, and literally get your molecule of DNA and it goes through a channel. So you feed this through a channel, it changes the current across the pore, the nanopore, and then that gives you the readout. So you can get really, really detailed information, but without disrupting the sequence. So all of our sequencing, we generally chop it up into little pieces and do sequence short bits, and then we map it back. So this can give us really important information about what we call structural changes. So sometimes segments of the DNA get put in different places and in the wrong order, and that can lead to cancer development. So we're applying these really exciting new novel sequencing approaches. So that's also some of the areas that we've been looking at. And then most recently, we've been looking at some splicing of RNA. So you have your DNA sequence that gets read, that makes RNA, which then makes the protein, these building blocks, the proteins that build everything in our body. You can have errors in how the RNA is put together. So that's called splicing. So we've been working collaboratively with colleagues in Spain, looking at some splicing of that. So we consistently look at all the different steps to try and see where something might be going wrong to make the tumor develop. So multifocal, the new long read sequencing, and then looking at some changes in the RNA as well as other developments since 2020.
That's so interesting. And actually, it made me think while you were talking that technology must be such a massive part of your work. And I guess it's probably quite an exciting time to be working in the field, because technology is developing so fast as well, isn't it? And it must be really integral to what you're doing.
Can I just briefly step in to put that into context? So I did my PhD around 2004, 2005, and with that was in bowel cancer. With that, it took me 6 months to sequence one gene in 100 cancers, and I was working hard. I wasn't sitting around. And to put that into context now, we can get a whole genome sequence, which is 3 billion base pairs, in about 72 hours. So the rate and scale, and in terms of the epigenetics and the DNA methylation, the area that I work in, it's been the most phenomenal time. It's been like being an explorer going out into space, because we're just learning things about the genome we never knew before. How some effects that are a long way away from the genes that are being read in the DNA, you can have changes, what we call upstream. So it's what used to be called the junk DNA. It's not junk at all. This DNA can then have an effect on how genes are turned on and off and how they're read. So it's been an amazing time for exploration, an amazing time for technology, the rate and pace. And what has had to evolve alongside that is our capacity for analysing data. And so bioinformaticians are absolutely integral for everything that we do. We wouldn't be able to work without them. So we're working very closely with data scientists and bioinformaticians to, so you might imagine you've got 3 billion base pairs, and you have, hundreds of reads of those 3 billion base pairs to make sure that that reading is accurate. And you're handling all of that data. So it's been the most phenomenal time.
And do you think, so we're hearing a lot about AI, aren't we, at the moment? Do you think that will come into play at some point in terms of kind of analysing large amounts of data? Do you think that could help?
Yeah, I mean, there have been some great examples where you can use AI or other tools when we're predicting. So I sort of mentioned you've got your changes in the DNA, then you have this RNA, then the protein. We've got really good tools now for predicting from the DNA change what the change in the functional protein will be, for example. So years ago, we'd actually sort of do that, rattling bottles of cells that were growing and knocking things out to see what would happen. But we have really good what we call in silico tools to then work out what those changes in the proteins are.
Wow, great. Okay, so let's move on, because we could probably talk about that for a while. We're going to be talking about clinical trials for question 4. So really, it would be interesting to kind of hear your thoughts on, some advantages and disadvantages of clinical trials, both for an individual and then also kind of a level, perhaps thinking about society in general and how they're assisting.
Yes, thank you. I mean, as an oncologist, clinical trials are absolute bread and butter of what we do, because we're always looking for better treatments, we're always looking for better outcomes, we're always looking for better treatments for our patients, and we will never stop looking for that. That's why I became an oncologist, because we're always looking to improve things. And the best and most robust way to do that is to have a very well planned out clinical trial where you can get some definitive outcomes and answers, which can then help us decide if treatment A is better than treatment B, or no treatment, or less treatment. So that's sort of the background and the bread and butter of oncology, although clinical trials and is applicable to all areas of medicine, absolutely all areas of medicine. I think on an individual level, I think starting with the positives, as being part of a trial, you get brilliant care and follow-up. You might get access to treatments which aren't currently available, or haven't been proven to be available. But the flip side of that is you might then be given one of the treatments in the trial which isn't superior or better than the standard of therapy. So there's always this, the juxtaposition of there could be an opportunity to receive a treatment that might improve that individual's outcome or not. So that's something to bear in mind. And I guess another downside for some patients could be that, you often need to come to the hospital more often than previously. So there's sort of this balance of what you need to invest personally in terms of your time. When people are given information about clinical trials or studies that they might, become involved in, there's a lot of information to go through, because quite rightly, we're obliged to let individuals know what some of the predicted side effects and what we call toxicities might be. And some of that can be a bit overwhelming. So it's our job to sort of help and support people, absorb that information and make their own decision as to whether it's the right thing for them to do. And the most important element of that is that if anyone enrolls in a clinical trial, they're not obliged to stay on it forever. If you feel, if it isn't the right thing for you, then you can withdraw. And it's not something we sort of advocate, obviously, because it's much better for everyone if you can, if we can actually get the data and the outcomes. But no one is ever pressurized to be part of that clinical trials. But I know, in my experience, in terms of the treatments we use in neuroendocrine cancer, the ones that are proven, and the sunitinib and the everolimus and certain types of chemotherapy, we wouldn't have had, the information to give those treatments without all of the patients that enrolled and supported those clinical trials. So it's such important work, really, really important work. And I suppose that then takes a step into, from the individual level to society. It's the only way that we can improve what we do and how we do it. And if we can learn that new approaches will be, beneficial to our patients.
And how do people get involved in clinical trials? Is it often through the team that they're working with at hospital, or could they kind of look things up online as well? I think there's kind of a centralized website for clinical trials.
So we have a group nationally, a neuroendocrine cancer, that it works across hospitals nationally to think about trials which might be upcoming and how we might host and roll those out. Usually the first port of call for a patient undergoing treatment, it will be their local team that might say, have you considered trial A, B, or C? If that individual hospital isn't actually running that study or that trial, it could be that that patient then goes to a different hospital or a different centre to get access to that trial. So because of that, we keep records nationally in the working group, and and we've been working closely with Neuroendocrine Cancer UK to keep things updated on the website around which clinical trials are open nationally as well. So I think the first port of call is your local team, but then people like NC UK and other websites and approaches can give you an idea of what trials are open nationally.
And if, so like you say, if somebody has to travel perhaps to a hospital a bit further away, is there kind of support to do that within clinical trials?
This varies from study to study. sometimes there is funding within the what we call, when you set up the trial and get the funding for it, there might be some money and funding available to support patients who have to travel to that trial to get that treatment. But unfortunately, that isn't always available. So that's something just to discuss and bear in mind, you know, if you're thinking about entering a trial, and discuss that with the local teams and the teams that are running the study.
Okay, so it varies. Okay, great. And so we're on to our last question. Really is just to, usually we kind of wrap up the Q&A sessions just asking, you know, are there any developments that you think are kind of worth mentioning to the neuroendocrine cancer community, you know, that might be of interest. But there was one specific one from your summit talk. So you talked about that there was an aim to incorporate genome sequencing alongside clinical trials. So I don't know if you could kind of expand on that a little bit as well.
Yeah, so in terms of the genomic medicine service and cancer as a whole, that sequencing is now routine. And historically, clinical trials, there might be extra money and funding within a clinical trial, for example, to pay for that sequencing for that one target that they're looking for that clinical trial. Whereas now we're moving towards and setting up where that sequencing is done as part of your NHS treatment, and then that can be used and shared to guide which clinical trials patients would be eligible for. So that's a step change. So we're working nationally. I'm on a national cancer genomics working group. So that's with the head of cancer for NHS England, Peter Johnson, and the chief clinician for CR UK, Cancer Research UK, Charlie Saunders. So we've got charities, we've got NHS, we've got all the people involved around how can we use this information and data most effectively to benefit our patients as quickly as possible. And that's one thing. So there's no point repeating if we've done the test already. It's about then sharing that so it can help guide patients into different clinical trials. And with the I know I did mention the summit talk, the Genomic Tumor Advisory Board. So that's something that's still in development, but it's something like a Genomic Tumor Advisory Board. You can say, well, you know, patient A on their sequencing, they might be eligible for trial A, B, or C. And let's think about that. So that's something that we're working towards being the sort of the bread and butter. So using that sequencing to help identify clinical trials for patients. And again, with that, we can link that up nationally. So if you're in the southwest where I live, it might be that there's a study that's appropriate in Birmingham, and it's whether then that it's right for that individual to then, you know, go and think about having a clinical trial elsewhere.
And then just any other thoughts kind of I can.
Yes. So thinking about so just sticking with clinical trials for a little while, there's been quite a lot of work recently and studies recently around, can we make radio-targeted therapy, so PRRT, more effective if we combine that with either our standard chemotherapy, so some of our chemotherapy drugs such as capecitabine, or molecularly targeted drugs, so everolimus and those sorts of drugs, to see if that makes the PRRT more effective. So far, there hasn't been anything sort of that's been a trailblazer coming out of that, but that's an area where there's still a lot of work in clinical trials ongoing. So I think I would say call that a watch this space. When we think about immunotherapy as a community, I'm sure you know you're all aware that in some other cancers, immunotherapy has had quite a big impact on subgroups of cancers. It's never, in any cancer, it's never that it's beneficial to every single patient. So you always have to identify that subgroup. There's been an enormous amount of work in neuroendocrine cancer, and sadly there aren't that many opportunities to use immunotherapy in neuroendocrine cancer. But again, there's still some ongoing work about using those drugs in combination with other therapies. So there's still some sort of unexplored avenues and answers we're looking for in that area. And then in terms of the basic science research, something that we've lacked and we still lack at scale is what we call models, so what we call in vitro models. So again, thinking about other cancers, you can take part of the tumor and then you can grow cell lines. So you have little pets of these cancer cells literally growing in flasks. And then if you've identified something in your DNA sequencing, you can manipulate that in your cell line and look for effects. And that's often been one of those stepping stones to then going on to maybe giving treatments in humans, or thinking about taking that process one step further. We've really struggled to have reliable models and cell lines in neuroendocrine cancer. There's been some movement in that area, but where there's been a really exciting development is organoids. And organoids are little groups of cells. So that's looks a little bit more like the actual tissue rather than just cells floating around in a liquid. And so there's been some really positive findings in developing organoids. So this came out of Hans Cleaver's group, and then Talia Dayton, she's recently moved to Barcelona, has a group working on organoids in bronchial, intestinal and pancreatic nets. And so I think that's been a real piece of progress to help us then test things and work out what approaches or treatments might be useful for our patients, and also to understand more about the biology. There's still a lot of work to do in that area, but it's a really positive step forward. Yeah.
Always more work to do.
Always more work to do. Yeah. Well, it's always a good reason to get out of bed in the morning.
Yeah. Oh, thank you, Chrissie. So yeah, it's really interesting kind of hearing your real passion for the subject. Is really, really interesting to hear. So yeah, thank you, Chrissie, and hopefully this video has been useful for those watching. And this Q&A session is part of a series where I speak to different medical experts covering a range of questions within neuroendocrine cancer care. So keep an eye out for those within the handbook. And thank you again.
Thank you. It's been a pleasure.